All right, so we're going to start today by comparing eukaryotes and prokaryotes. So here is the screen. We're all sharing it together. You can either listen and write stuff down in a t-shirt if you'd like, or you can just watch, right? So in terms of the differences between eukaryotes and prokaryotes, the first thing that they're going to ask about is the DNA. So let's see if we can make this a little bigger. No, never mind. Okay. So the DNA in a eukaryote, remember, is a couple things about it. It's enclosed in a nucleus, right? So it's separate from the cytoplasm. That's one definite difference that you need to mark. Here, there's no nucleus, right? No nucleus in a floats in the cytoplasm. That winds up being a rather important difference. We'll get to that soon. Okay. Enclosed in the nucleus. It's organized, the eukaryotic DNA is organized into linear chromosomes that incorporate histone proteins, right? There's lots of packing in a eukaryotic chromosome. DNA is wrapped tightly around histones, and a lot of times that's going to definitely affect whether it gets expressed or not. In prokaryotes, no histones, and DNA is circular. Circular chromosome. Very different from a eukaryotic. Okay. Into linear chromosomes, circular chromosomes. There we go. Okay. Okay. Replication in eukaryotes versus prokaryotes, it's about the same. You need DNA polymerase, and other enzymes. Okay, and they're really pretty similar. You can copy and paste this for the most part. About the only thing that's different, you have multiple enzymes. You have, in this one, you have, replicating a linear chromosome is going to be a little bit different than a circular one, right? But you're going to have replication forks going in different directions, and you're going to need to replicate all of it, and it all happens, occurs during interphase, which is the same in both. Right. Interphase. Oops. Okay. All right. Not a whole lot of differences there. Okay. Transcription. Transcription in eukaryotes. Which enzyme do we need? A one-stop shop enzyme that does transcription of a eukaryotic gene. RNA polymerase. Okay. Oh, I'm over here. Same enzyme. Promoter is present for both. That there's a lot of differences. Need transcription factors bound to promoter. Here. controlled by. Say controlled by. Repressor proteins. Okay. I'm going to add something to the prokaryotic DNA up here. Genes organized into operons. And over here, no operons. Some of this isn't going to make sense until we finish the notes today. I'm not going to give you all of it, but transcription and in eukaryotes versus prokaryotes, there are lots of controls over here on both, really. So you need transcription factors bound to the promoter. And you also need the chromatin to be unwound. And over here you have no chromatin, right? No histones, so no unwinding necessary. Okay, that's good. All right, post-transcription. Yes, messenger RNA gets edited for translation. No, mRNA is not edited, not in bacteria. Am I going too fast? You guys with me? I mean, I will post this later, but I just thought, for some of you, I know it's nice to write this and type this along with me, so hopefully it's helping. Okay. All right, keep going. Translation. The translation of these is in a eukaryote occurs in ribosomes in the cytoplasm, completely separate from the transcription step. Right. I'll give you a minute to absorb that one because that's an important one. Translation, you learned by coloring in the big diagram, right? That the transcription happens in the nucleus and all the editing, and then the RNA goes into the cytoplasm, swims over to a ribosome, and the translation happens there. So they're very, very separate steps. The prokaryotes, the bacteria, as we're going to learn, translation and transcription often happen at the same time. There's no separation of these steps. Big difference. Okay. All right. In the eukaryotic cell, yes. polypeptides can be altered after translation. You've got the ER, you've got chaperonins that do some folding, you've got a Golgi body that does final processing, you've got lots and lots of areas and special chemical compartments in a eukaryotic cell that can do lots of fiddling with a protein. Not as much in a protein, not as much in a bacteria. The protein you finish with is pretty much the protein that you get. Not quite as complex. Okay. So hopefully that kind of wakes up some memories from freshman year and may also help flesh out what we learned last time when we were learning about eukaryotic gene control. All right, I'm gonna